reason why I chose human brain organoid or mini brain as the topic of this documentary is because I recently was volunteering at a senior care center and I met friends who had different challenging mental health experiences like autism, seizure, Alzheimer's disease, neurological damage, or disorders. We want to know what caused their illnesses and if there is a cure for them. The recent advancements in developing a miniature brain using human stem cell cultures in a laboratory environment will help us understand how our human brain develops from a single cell and how certain neurological illnesses begin and progress. The goal is to better understand the pathology of neurological diseases and find treatments or cures for them. Mini-brains, or human brain organoids, are the latest development in stem cell technology, which started gaining widespread attention since 2008. Mini-brains, or cerebral organoids, are organized 3D cell structures made up of different brain cells, which resemble the complexity of a human brain. Human stem cells can be grown into brain organoids in a culture or on a dish stored in a laboratory. This offers neuroscientists an unprecedented opportunity to more accurately model the human brain development and disease. The human brain is one of the most complex, mysterious, and beautiful organs that controls all the functions of our body. It receives and interprets information through our 5 senses. It controls our thoughts, memory, and speech, the functioning of many organs within our body, and the movement of the arms and legs. The brain has 3 main parts: the cerebrum, cerebellum, and the brainstem, with each performing different functions. The wrinkly outer layer of the cerebrum is the cerebral cortex, which is the largest part of the brain, controlling higher-level functions like cognitive functions, memory, and speech. There are approximately 100 billion neurons in the whole brain. Neurons are highly integrated nerve cells and are organized into columns and mini-columns that make up the many layers of cells within the brain. Neurons are connected by trillions of connections called synapses. Neuron cells pass chemical and electrical signals along the pathways in the brain. This is how information and messages get transmitted to and from the brain throughout the body. Information from one neuron flows to another neuron across a small gap called a synapse. At the synapse, electrical signals are translated into chemical signals in order to cross the gap. Once on the other side, the signal becomes electrical again. This is the process of neurotransmission, which enables signals and information to travel throughout our body. It is easy to visualize the brain as being an electric powerhouse containing bundles of electric wiring generating electricity. Our brain is indeed a beautiful and amazing organ. A simple movement like serving or hitting a tennis ball, for example, involves a complex interaction of different parts of the brain commanding the executive functions from the cerebrum, like judgment, planning, reasoning, memory, recollection, and precision. It also involves the cerebellum to coordinate muscle movements, maintain posture, and balance. At the same time, the brain stem controls blinking, breathing, heartbeats, and body temperature. Every part of the brain is involved in this simple movement. So with this basic introduction on the human brain and neurotransmission, so what is exactly a human brain organoid? Organoids are 3D biological structures, essentially a cluster of cells constructed from stem cells growing in a culture or dish in a laboratory. Stem cell-derived organoids partially mimic the functions of real organs. Today, different types of organoids have been developed, like cardiac, kidney, lung, and cerebral or brain organoids. There are two main varieties of organoids depending on the cellular source, either pluripotent stem cells or somatic stem cells. Scientists used to develop two-dimensional stem cell culture in the lab in order to study biodevelopmental issues or different organs. Now, using a three-dimensional cluster of cells mimicking the real organs, the 3D human brain organoids are a very useful model, giving us new insight to how different parts of the brain develop, function, and communicate with each other. More importantly, 3D human brain model helps neuroscientists understand the early development of brain-related conditions, causes of mental illness, and the possibility for finding targeted therapies in the future. The technological advances of developing the three-dimensional mini-brain started in 2008 and went through interesting and amazing breakthroughs. Scientist Dr. Nicolas Gaspard of Belgium published a paper in 2008 describing a natural mechanism of cerebral cortex development through the coordinated generation of different types of neurons using embryonic stem cells taken from mice. Further stem cell research showed that human-derived embryonic stem cells can also spontaneously form pattern structures mimicking the early aspects of development in the human cerebral cortex. In 2013, Dr. Madeleine Lancaster of Cambridge of the UK created the first human brain organoids from human pluripotent stem cells, which have the capacity to recapitulate specific features of the human cortical development. Since then, other scientists have worked to develop a more complete form of the human cerebral organoids. They began to grow deep and superficial cortical layers of neurons. Some of these, more advanced organoids exhibited spontaneous activity corresponding to a 19 to 24-week embryo. Other scientists developed different parts of the brain with specialized cells in the organoids containing all 6 cortical layers of the human cortex. Other developed cerebral organoids were able to generate nerve tracts with functional output with projections extending outside the organoids. The organoid stimulation was shown to cause muscle contractions. New co-culture techniques were used to combine various brain regions with one organoid tissue, allowing complex interactions between different brain regions. Mature inhibitory and excitatory neurons were later identified in the long-term cultured cerebral organoids induced from H9 human embryonic stem cells. It was found that organoids developed over an extended period showed advanced features such as spontaneously active neuronal networks and functional photosensitive cells. Recently, in March 2019, Dr. Madeleine Lancaster, who led the work at the Medical Research Council's Laboratory of Molecular Biology in Cambridge, developed a miniature brain in a dish with a spinal cord and muscles attached. A little blob of human brain cells were seen to spontaneously send out tiny connections to link up with the spinal cord and muscle tissue which was taken from a mouse. The muscles had been seen to visibly contract under the control of the attached brain organoid. This latest blob shows similarities in terms of the variety of neurons and their organization to the human fetal brain at 12 to 16 weeks of pregnancy. This summary shows just how fast the rate of advancement is in stem cell research and mini brain development. These experiments show us how our human brain and nervous system develop, and they help us understand the onset and progression of brain diseases. These advances will ultimately help scientists find the right cures for mental illnesses and brain damage. There are many important implications for our society as a result of these revolutionary technological advancements. They include longer lifespan for people, especially those affected by neurological diseases, much improved quality life of patients, possible early detection for diseases, more effective treatment or cure for mental illnesses or developmental disorders, a better rate of full recovery for brain trauma patients, and ultimately, a more efficient allocation of medical care resources. It is amazing to realize that stem cell research breakthroughs will mean that our brain, which has always been known to be the hardest organ to study, will no longer present a challenge to scientists in understanding its developmental biology and pathology. It is hoped that within ethical limits, stem cell research and neurology will advance fast enough so that pre-existing patients or people carrying problematic genetic makeup will benefit from the right treatments or preventative measures.